Hello students, welcome back to my biology online classes. In my previous two lectures you had already known that I started developmental biology, which is under semester 6 CC 13 University of Calcutta syllabus. Today I am going to discuss with you about spermatogenesis. As the name suggests, genesis means to generate something, and the prefix spermato means sperm. So the process spermatogenesis I am going to discuss with you today is about how the sperm is generated. Process of generation of sperm. Okay, sperm is male gamete which is produced in human male body. Where is the location of sperm production in human body? The location, a place within seminiferous tubule of testis. Each testis has about 250 compartments called testicular lobule. Each lobule contains 1 to 3 highly coiled seminiferous tubule. Each lobule contains 1 to 3 highly coiled seminiferous tubule. And here in seminiferous tubule, sperms are produced. Each seminiferous tubule is about 30 to 70 centimeter long and almost 200 micrometer in diameter. And here between the tubules, another important terminology you should know prior discussing spermatogenesis, that one is Leydig cell. Between these seminiferous tubule, Leydig cell, the tubules. Why the Leydig cell is so important? Because Leydig cell produce male sex hormone testosterone. Male sex hormone testosterone. And this testosterone hormone, it has a great effect on this process spermatogenesis. So prior to discuss the actual mechanism of spermatogenesis, you should clear about these conception. Okay, each day about 300 million sperms complete the process of spermatogenesis. Each day about 300 million sperms per day complete the whole process of spermatogenesis. Actually, in spermatogenesis there are completely 3 steps. It needs 3 steps to complete a whole round of spermatogenesis. 3 steps: mitotic cell division or phase of multiplication. Number 2, meiosis or phase of maturity. And the third stage, spermiogenesis or metamorphosis of spermatid into sperms. What are 3 steps? First steps is about mitotic cell division. Next step, meiosis cell division. And the third and last step is metamorphosis of spermatid into sperm. In my previous lecture, I had already discussed with you in my second lecture about gametogenesis. I had already mentioned primordial germ cell. At the time of embryo formation, the primordial germ cell originated another place other than gonad, and the male and female gonad were not differentiated at that time. At the time of birth, primordial germ cell can migrate from yolk sac to testis during early embryonic development and become spermatogenic stem cell. That stem cell which can give rise to sperm when needed. Now, this primordial germ cell is termed as spermatogonial cell within the outer region of seminiferous tubule. What is seminiferous tubule? I had already discussed with you in my this lecture. The phase of development, mechanism of division, type of germ cell, germinal epithelium cell, and chromosome number. At 12 to 14 years. The 12 to 14 years in case of human male, it's the time when adolescence start, means puberty start. Okay? At the time of birth, then when it enters testes, then at the time of puberty. Up to 12 to 14 years, we can divide this mechanism of division into three phases. At the time of birth, what was the situation? Then when entered the testis, what was that situation? And now at the time of puberty, what change will occur? Got it? At the time of birth, type of germ cell. At the time of birth, primordial germ cell, before birth, primordial germ cell from yolk sac can migrate to testis and enter testis. After that, at the time of puberty, what happened? It is the scenario at the time of puberty, known as primordial germ cell and the chromosome 46 means 2n. Now see the next stage, multiplication, mitosis. Here repeated division will occur, and here some of these type of spermatogonial cell can give rise to stem cell. About this topic I am going to discuss later with you in this lecture, obviously. Now known as spermatogonia, and the chromosome number is 46, and these are now known as primary spermatocyte. Here the chromosome number is 46. After that, from here meiosis division will occur. Meiosis division will occur. Secondary spermatocyte. And here meiosis division occurs and the chromosome number become half n. Okay? Next step, this one spermatid. And here the chromosome number 23n, and this spermatid, this can give rise to patent tail, and by means of metamorphosis, this one is now known as sperm. It's also 23N. Now write here, this here meiosis, here meiosis 2, and here, and this one is known as spermatogenesis, growth, and from here meiosis occurs means reduction of chromosome number, and here metamorphosis. So this is the whole process of spermatogenesis. Let's concentrate very carefully about this diagram. This diagram is the main center of attraction of spermatogenesis. In your exam, in your syllabus, in your exam, this one is your ultimate answer, this one is your ultimate target. Okay, what have we already discussed? There are 3 phases. First phase is about mitotic cell division, and the journey started from when primordial germ cell enters gonad and become spermatogonium cell. These spermatogonial cells about 12 micrometer in diameter. 12 micrometer in diameter. At the time of puberty, these spermatogonium cell divide several times by means of mitosis cell to produce a large number of spermatogonium. Some of them continue as stem cell. This one is stem cell, and this stem cell cannot participate the whole process, and this one is called type A spermatogonia, and this one differentiated into progenitor cell, and this one is called type B spermatogonia. It can give rise to other type of cells and can give rise to this progenitor cell. Type A spermatogonia serve as stem cell for spermatogenetic lineage, whereas this type B spermatogonia are the precursor cells and differentiate into sperm. The type B spermatogonia undergoes changes by accumulating nourishing materials obtained from the germ cell and almost become double in volume. This growth requires synthetic activity that are preparatory for the initiation of meiosis, and such cells are called primary spermatocyte. The primary spermatocyte is large, diploid or 2N. All are here 2N. Okay, possesses total 46 chromosomes. Then it enters into meiotic division. First meiotic division. Look at here. Meiotic division 1, your first meiotic division. What happened in first meiotic division? All of we know about meiotic division. The first stage of meiotic division is what? Prophase, and prophase has leptotin, zygotin, pachytin, diplotin and diakinesis. Prophase is followed by metaphase, anaphase and telophase. To complete reductional division 1, meiosis 1. The daughter cells produced at the end of meiosis 1, they are smaller and termed as secondary spermatocytes. They are haploid. From here, they are haploid and the chromosome number is 23. The secondary spermatocyte undergo meiotic division 2. The single cell produce 2 daughter cells, and and they are now known as spermatid. Here also n number of chromosome and total chromosome number is 23. And after that, look, from each spermatogonia, each type B spermatogonia, 4 spermatids are generated. The next step is very much important, because the next step is not about meiosis division, not about mitosis division. It is metamorphosis. We know about metamorphosis when at the time of growth and division, when the abrupt change of structure occurs, then it is known as metamorphosis. And in this case, from which they metamorphose and which product will be generated, they are completely different. They are completely different morphologically, different each other. Totally dissimilarities are shown, as we already study in metamorphosis in tadpole larva and the mature frog does not have any similarities. So here metamorphosis and differentiation occur and the spermatid changes into mature sperm, and the step is known as very much important step. Very much short questions are coming from this part, and this step is known as spermiogenesis, and this stage is also called spermatogenesis, and the release of sperm is called spermiation. So each spermatid differentiate into mature, also called spermatozoa, and this differentiation means these include formation of acrosome, condensation and elongation of the nucleus, development of the flagellum, and the loss of reductant substances of the cytoplasm and its organelles. Thus, 4 sperms are found from single spermatogonium cell. After spermiogenesis, what happens? After spermiogenesis, sperm had become embedded in Sertoli cell and finally released from the seminiferous tubules by the process called spermiation. And what's about Sertoli cell? Here I have used a term, Sertoli cell. Previous semester, semester 3, you had gone through histology of testes, and at that time you have found about Sertoli cells. In spermatogenesis, Sertoli cell, these Sertoli cells play a vital role. That's why I am going to mention some of its important functions. Sertoli cell supply the nutrition to the sperm and reproductive cell. What is the second function? Some of the degenerated cells of seminiferous tubules are destroyed by phagocytosis. It play a role in secretion. Point number 4, help in steroid hormone secretion, help to release sperm in the lumen of the tubule, and fifth one, helps in spermiation, means helps in release sperms from seminiferous tubule. Within the release of sperm in the lumen, the process spermatogenesis come to an end. So this is in a nutshell the process of spermatogenesis. Here also an adding, which part of the spermatogenesis is not required here, but in case of in the process of spermatogenesis, the role of hormones in spermatogenesis is very much important things. So I am going to discuss that in a nutshell also. So come to the last point. In developmental biology, the hormonal control of spermatogenesis is not so much needed, but without the hormonal control of spermatogenesis, the lecture of spermatogenesis and knowledge of spermatogenesis will remain incomplete. So first discussion about hormonal control of spermatogenesis. All of we know, here is a chart. All of we know about hypothalamus. Hypothalamus is situated in our brain. I'm not going to discuss about hypothalamus here. It's not about hormonal control class. It's just about hormonal control on spermatogenesis. So hypothalamus releases gonadotrophin release hormone that will act on anterior pituitary. All of we know about anterior pituitary. It is known as the master gland From anterior pituitary, 6 trophic hormones are released, which has immense effect all over our body. I am not going to discuss all of them. I am going to focus 2 trophic hormone, LH and FSH. Full form of LH is luteinizing hormone. Full form of FSH is follicle stimulating hormone. Out of those 6 trophic hormone, here this 2 hormone is needed for spermatogenesis process. Both LH and FSH hormone acts on testes. Here all the scenario inside the box. This is assume this is inside testis, and effect of LH and FSH inside testis, human testis. Exert its effect on Leydig cell to secrete testosterone hormone. On the other hand, FSH says give instruction to Sertoli cell just to help for spermatogenesis. On the other hand, testosterone also exert its effect on Sertoli cell and spermatogenesis. Okay, positive feedback. So all the process is on single side. Do secret, do some work. That's why positive. Hypothalamus says do your work. Pituitary, I am giving you GnRH, positive. So all are these positive feedback. LH give positive signal to Leydig cell, that's safe to secrete testosterone. FSH send its positive signal to Sertoli cell to help spermatogenesis. Testosterone also send positive signal to Sertoli cell to help in spermatogenesis. Okay. Now negative feedback is when needed when too much testosterone hormone is secreted. Testosterone hormone also helps the secondary sexual characters in males at the time of puberty. Yeah, all of its sexual age of a male. Okay, so when too much testosterone is secreted, all of we know about our conception about hormone. Hormone is needed in an optimum certain amount, not hypo, not hyper. Not too much is needed, not too less is acceptable. Okay, so negative feedback is necessary when excess hormone is generated and the process is continued. So testosterone, the amount of testosterone is high now and it is not necessary, it was too much level. So testosterone exerts its negative feedback effect on anterior pituitary. Here I am giving negative sign. It says anterior pituitary not to secrete any more LH and FSH. Stop signal, negative signal, and says anterior pituitary please stop secretion of LH and FSH. It is not needed now. The level is too much high. It's also saying its negative feedback signal to hypothalamus just to say, convey a message to hypothalamus not to secrete and furthermore GnRH on anterior pituitary. Okay, so this is negative feedback of testosterone and this is positive feedback. So this is in a nutshell hormonal control of spermatogenesis. In the next class I am going to discuss with you the detailed structure of a mature mammalian sperm and its every details. Hope all you have enjoyed this session of spermatogenesis. So stay connected, stay tuned. The next class will be about structure of sperm.